Hello there. This is Dan Guerra, and I'm once again coming to you from VerEvMed, where you can interact with scientists who will verify scientific evidence from the medical, physiological, pharmaceutical, microbiological literature. What these YouTube presentations are supposed to be about is for me to kind of demonstrate the sort of things that we do when we discuss in depth what we find in the literature after questions are posed to us from clients. So what I'm trying to show you is what we might do if someone were to ask a more general question about pharmaceutical targets for, in tonight's discussion, major depressive disorder. If you want to interact with us on a one-on-one basis, wait to the end of this presentation. You will see where you can contact us via our website or via email. So I'm going to go ahead and get started right now. And here we go. So there's our logo. So again, tonight I'm going to talk about major depressive disorder. My name is Dr. Dan Guerra, and this is March of 2017. There is a new DSM5 criteria for major depressive disorder, which I'm not going to display here tonight, but you can go to that link and you can find out all of the specific designations of the disorder. It hasn't changed substantially since DSM-4 in terms of the general criteria, but there are several secondary criteria, I think, that have been modified. So if you're interested in that, I suggest you go to that website. Okay, I'm going to start off with talking about a paper. This is a very just brief discussion of it. And this comes from Neuropsychiatric Disease Treatment. This comes from 2016, late last year, approximately 3 months ago. And just to briefly describe what the paper talks about, it tells you that major depressive disorder is probably the leading neuropsychiatric disorder, and that by and large, it's still treated as one that is involved in monoamine neurochemistry. Now, let me mention here before I continue, this particular YouTube is for professionals, people that already have an advanced doctorate degree or people that are working towards that advanced doctorate degree or who have otherwise a lot of training in the area. We also work with clients that do not have that sort of advanced education, including people that have degrees, bachelor's degrees, or even people that have not gone to college. So we gear our discussions accordingly to who's asking the questions and what they're actually looking for. So this talk tonight is more geared towards our friends who are physicians and people who have otherwise terminal degrees. So monoamine neurochemistry is considered the most major target in MDD. MDD is diagnosed in both sexes and in many age groups, although it's probably more often treated in younger adults. In the U.S., almost 16 million people, or that is about 6.7% of adults, have reported having one major depressive disorder episode sometime in the last year. It's the most prescribed drugs that are used for MDD are ones that are targeting the monoamine system, and this usually is associated with serotonin reuptake inhibition or serotonin and norepinephrine reuptake inhibition. And I will describe those and show some detail later what I mean by that. In the study that was described in this particular publication, this published refereed journal article, they were really looking at adherence to prescription. So a fair amount of the research in this area is looking at if drugs are prescribed, are those drugs adhered to by the patients, or do the patients stop taking the drug? So that's what this particular paper was about. And what they determined was that there was one drug, desvenlafaxine, which is an SNRI, which was the most adhered to drug of the ones they examined. And they examined all the standard SSRI treatments. So that was what this paper was about. Not anything earth-shattering in that particular publication, but there are people interested in knowing these kind of clinical trials and whether or not the drug that they may prescribe is the one more likely to be adhered to. And a lot of reasons for that, behavioral reasons, probably some neurochemical reasons as well, which we would find in other aspects of the literature, of course. I want to mention here, though, at the beginning, at the outset, that we've been talking right now about monoamine therapy, And I would want to make it clear that that is not the only target. That's not the only neurochemical axis of which to discuss major depressive disorder. And we're going to see that as we go on this evening. This is a slide just showing you, it's a sagittal view of the brain, midline sagittal view, and it's showing you what are some of the nuclei or regions of the brain that have been mapped to MDD, basically many regions, not just the limbic region, but also areas in the prefrontal cortex. So here we're looking at the amygdala, the hippocampus, the brainstem, the callosum and the corpus callosum, the dorsomedial frontal cortex, the midcingulate cortex, orbital frontal cortex, the prejudial anterior cingulate cortex, posterior cingulate cortex, and the medial frontal cortex. So you can see it's scattered throughout. So when MDD is examined at the brain level, where in the brain do we think that there are nuclei and there are neurotransmitting systems, synapses, and neurons, and probably glia that are linked to MDD? It's basically in many regions of the brain, not in one specific location. And that, of course, is going to give you different interpretations from the different aspects of the literature. So what else can we say? We can say that there are abnormalities that have been reported in certain anatomical regions of the CNS: prefrontal cortex, the basal ganglia, the hippocampus, thalamus, cerebellum, and indeed in the temporal lobe. In individuals suffering from unipolar depression, the most reliable findings are found in the PFC, that's prefrontal cortex, ganglia, hippocampus, and cerebellum. And there is also the possible relationship between the hippocampal volumetric reduction that seems to happen with age and the HPA axis, that's the hypothalamus pituitary adrenal axis, which we'll get into later. And that's going to involve more than just the monoamine circuitry. What we can say right now is that this HPA axis is correlated closely with glutamatergic theory of MDD. So rather than the monoamine, which describes the catecholamines, particularly serotonin, norepinephrine, and dopamine, there is also a theory on what is the major axis of MDD in humans, and that involves glutamic acid neurotransmission. So I want to bring that up now. A lot of writing, let me go through it quickly. Familial cases of unipolar and bipolar depression show an abnormality in the PFC. Some studies have found altered volumetric measures that we talked about. Post-mortem studies have found a reduction in the number of glial cells in that region. The PFC has extensive connections with, of course, the limbic system. That would be the amygdala, nucleus accumbens, hypothalamus. And there we find the neurochemistries of norepinephrine, 5-HT, that's 5-hydroxytryptamine, also known as serotonin, and DA, which is dopamine. Functioning imaging studies have also reported abnormalities in function. Increased activity may occur in some areas of the prefrontal cortex, amygdala, and the anterior cingulate. And consistent with the literature that suggests that sadness and anxiety disorders increases activity in those areas of the PFC. Those increases in activity by doing neural scanning are also localized to the ventral, orbital, and cingulate cortices, and they may be related to quite an array of emotions, and indeed some obsessive compulsive ruminations. It's an artist's depiction on the left showing you what you see on the right, which is a synapse between two neurons. When we talk about reabsorption, of course, what we're talking about here are the catecholamines or the monoamines. So serotonin itself is synthesized from the amino acid tryptophan, an aromatic amino acid. So you make 5-HT, which is serotonin. It gets packaged into the presynaptic neuron. Then it gets sent out into the synapse. So these different colors of these chemicals here are supposed to show you that not only do you have serotonin, you also have norepinephrine and dopamine. So what we're talking about when we talk about drugs used for major depressive disorder that are specifically targeting monoamine pathways, we're talking about blocking the reuptake of those monoamine neurotransmitters in the synapse. When you block reuptake, it's more likely that you're going to get binding of those monoamine neurotransmitters like serotonin to the serotonin receptors, which are going to be on the postsynaptic neuron shown down here. So those are the receptors that are going to bind to your neurotransmitter. And then you're going to signal into that postsynaptic neuron, and you're going to signal through more serotonin, norepinephrine, dopamine, acetylcholine, GABA, and in fact, glutamate and glutamine. All right. So once again, talk about regional CNS neurotransmitter distribution, specifically talk about the monoamines. The PFC has all three. The amygdala has all three. The nucleus accumbens does not have norepinephrine. Hypothalamus has all three. The striatum has only serotonin and dopamine, or no norepinephrine. And cerebellum, serotonin, and norepinephrine. So there's a lot of distinction between specific regional locations in the CNS. Now, the striatum is a convergent point for also the glutamatergic inputs from the cortex and from the thalamus, as well as the dopaminergic afferents from the midbrain. So this is where there's going to be an axis of interaction between the monoamines and glutamic acid and all of its possible annotations associated with its many receptors. This is the source of the direct and indirect pathways described in neurochemistry and in neuroanatomy, neurobiology. So you have two parallel basal ganglia circuits that are critical for motor function as well as for procedural learning. So that's where the striatum really comes into play. And the striatum also is a location in the CNS that's associated with obsessive compulsive disorder and with many forms of drug addiction. Here's this brief discussion of a paper that was published in 2015. The CNS anatomical regional specificity of that links cognition and mood are innervated with monoamine neurotransmission, including, as we've been saying, the tryptophan derivative serotonin, and then the tyrosine, another aromatic amino acid, metabolites NE and dopamine. Okay? So there's crosstalk between all of those neurotransmitters, and that crosstalk can be agonistic, that means supportive, or antagonistic, which means, of course, a diverse interaction, or that is polar interactions. The ligand activation of the 5H2A and 5H2C, those are the designations of those just two receptors, actually inhibit postsynaptic norepinephrine and dopaminergic neurotransmission. D2-like receptors promote 5-HT neurotransmission. So that's the adrenergic D2, the dopaminergic receptors. Presynaptic alpha-2 adrenal receptors actually antagonize serotonin signaling. And postsynaptic serotonin receptors, shown there, modulate GABA interneurons, which of course is inhibitory neurotransmitter, not a monoamine. And then subsequently alter 5-HT, acetylcholine, NE, dopamine, and glutamate neurotransmission. So as I said, a great deal of crosstalk. What kind of drugs are we talking about that are commonly prescribed? I'm sure you're well aware of these, but again, the ones that we're going to talk about at the get-go here are going to be the monoamine targeted pathway ones, SSRIs, citalopram, reboxetine, sertraline are common. SNRIs, which are also going to be affecting the neuroepinephrine axis, are going to be duloxetine and venlafaxine. NDRIs, the major one that's being prescribed is bupropion. Then we still have old school TCAs, those are tricyclic antidepressants, which are sometimes still prescribed, but those are less selective. And then you have the alpha-2 adrenergic antagonists as well, as sometimes find in the pharmaceutical or in the pharmacopoeia. What about new drugs for MDD? This comes from a paper published just about a year and a half, almost 2 years ago now, I guess, about a year and a half ago, in Progress in Neurobiology, citation shown there at the top. Here we're moving away from the monoamine pathway. We're looking at the glutamatergics. So there are multiple forms of drugs here. They're the non-competitive NMDA receptor antagonists. Major one that's being looked at right now is ketamine, which is also, of course, a drug that's used recreationally at higher doses, of course. There's also subunit specific NMDA receptor antagonists shown there. There are NMDA receptor glycine site partial agonists, because glycine has to work as a co-ligand at some of those glutamatergic receptors. There's also the metabotropic glutamate receptor, and there are drugs that modulate that. We say modulate because we really can't say it's antagonistic or agonist. It's a modulation, which hopefully we'll get into a little later what I mean by that. And then we have glutamate receptor targeted drugs directly, which also involve the alpha amino 3 hydroxyl 5 methyl 4 isozazopropionic acid, or AMPA agonists, and the mGlu-R2 3 negative allosteric modulators. So you can see there's quite a pharmacopoeia of drugs there working on the glutamatergics. Not to leave out, but there are also drugs targeting acetylcholine. There are also drugs that target the opioid receptors, which I have coined the term opionergics. I don't think anybody else has called it that. I've decided to call it that. And then there's also the corticotropinreleasing factor, releasing hormone receptor antagonist, and we'll talk a little bit about that later as well. Now, besides all these drugs and all these targets that are specifically for neurotransmission, there's also non-drug alternatives in MDD. Not only is there cognitive behavioral therapy, but there's also a nutritional arm of MDD therapy, and that involves caloric restriction or fasting. Brief background of some of these other neurotransmitters, which we're just going to kind of like race through. GABA, gamma-aminobutyric acid, is the chief inhibitory neurotransmitter. Plays an important role in regulating neural excitability throughout the entire nervous system. If you have disrupted GABAergic signaling, it has been implicated in many kinds of neurological and in psychiatric pathologies, including movement and anxiety disorders. So you see some of this in lots of major neuropsychiatric diseases, also in epilepsy, schizophrenia, and in drug addiction. Because I'm a biochemist, I like to show biochemical pathways. Sorry, there aren't structures here. I would prefer if there were, but we don't need to do that right now. But coming directly from the TCA cycle, or affectionately known as the Krebs cycle, for some of us who have known it for that long, from alpha-ketogluteric acid, 2-oxoglutarate shown here, you can synthesize glutamic acid via aminotransferases. Okay, aminotransferases will take an oxo acid and convert it into an amino acid just by adding an amino group from either another amino acid or directly from ammonium. Likewise, you can oxidize glutamic acid, and when you do that, you remove ammonia, shown here. This is glutamate dehydrogenase, a really important enzyme in converting glutamate directly to keto acids that are directly used in the TCA cycle. And the reason they're using the TCA cycle is for energy. So glutamic acid, being a very important neurotransmitter, is also directly linked to energy in neurons, in glia, and in the rest of the body, of course. Notice that glutamate is the precursor to GABA. So all you need here is a decarboxylation of glutamate, and you make GABA. Then GABA can be meet up with another amino transferase or transamination reaction, go back up to make succinyl semialdehyde, and this can be oxidized to succinate, and you're back into using that carbon directly in the TCA cycle, for example, to either reform malate NOAA and therefore run the cycle over, or to be gluconeogenic, or indeed to ultimately make ATP through the production of NADH. So what's the HPA axis? And we talk about that quite a bit as well in neuroscience. That's the hypothalamic pituitary adrenal axis, of course. It's also known as the limbic HPA. It's a complex set of direct influences and feedback interactions among the hypothalamus, pituitary, and adrenal. Those interactions are among those organs constitute that axis, the major part of what is known as the neuroendocrine system, So that's going to involve endocrine hormones, of course. And that's going to that entire HPA axis is associated with stress response in humans, and it regulates many body processes, including digestion and the immune system. It, of course, is directly associated with mood, emotion, sexuality, energy storage, and expenditure. So it's interconnected with the CNS. HPA in detail, key elements are the paraventricular nucleus of the hypothalamus, which contains the neuroendocrine neurons that synthesize and secrete vasopressin and corticotropin-releasing hormone or corticotropin-releasing factor, CRH, CRF. Those two peptides regulate the anterior lobe of the pituitary gland. Particularly, CRH and vasopressin stimulate the secretion of ACTH, and that's also known as corticotropin. That in turn then acts on the adrenal cortex, which produces the glucocorticoid hormones, and those glucocorticoids then also function to feedback regulate the corticotropin releasing hormone. So you can see that this is highly related. The neuroendocrine system is highly interrelated, ultimately to the production, because it's moving through the adrenal axis, of some of those very monoamines we talked about a little while ago. A little bit about corticotropin releasing factor or hormone, because you don't hear much about this in major depressive disorder, but it is actually quite important and vital in research areas. It's one of several neurohormones synthesized specifically in the hypothalamus, and it's released into the portal system, and it bathes the anterior pituitary. Right? So it's part of this HPA axis. It has marked CNS effects by acting at higher centers in the brain, including cortical regions where there's widespread distribution of receptors for CRF. The major role of CRF seems to prepare the organism for an appropriate response to various stressors, such as physical trauma, insults to the immune system, and in fact, indeed, to social interactions. It is the hyper and hyposensitivity of that system. Okay, so the teeter-totter effect is very common in neuroendocrinology. So that either hyper or hyposensitivity of the system that can lead to human pathologies, such as anxiety disorder, depression, such as MDD, and in fact, some feeding disorders has been linked to. So it's a very important axis, the CRF. So these results suggest that there is an excitatory influence of the CRF in the medial PFC, that's the prefrontal cortex, on stress-induced HPA activity and anxiety-related behavior. This all tends to indicate that the dorsal and ventral aspects of the prefrontal cortex may differentially regulate neuroendocrine and indeed autonomic paraventricular nuclei outputs in response to stress. So I know that's quite a mouthful, but I want you to get the full range of understanding of the complexity of the CNS. We talk about something like major depressive disorder. This is often overlooked or under-described. And certainly these are all very important aspects when you're thinking about targets for MDD. So what's the pharmacotherapy for MDD? Again, we said predominantly monoamine right now. Now that's despite of quite an ambiguous efficacy and a very large placebo effect. The placebo effect is its own neurochemistry. And in some subsequent VerEvMed mini lecture, I will talk about the placebo effect. Placebo effect is not mysterious, neither is it hyperbole. It is a real effect in the body, real effect in the brain, and we'll talk about it some other time. But placebo effect often outdoes some of the drugs that are targeting the monoamine pathway for MDD. So it's something you have to keep in mind. The new therapy then that have been talked about more in the last 3 or 4 years in the literature is glutamate neurotransmission. Of course, glutamate is the central excitatory neurotransmitter, and GABA, of course, is the inhibitory. Glutamate seems to be linked to MDD. The N-methyl-D-aspartate or NMDA receptor actually binds to and is activated by glutamate as well as glycine as a co-adapter amino acid and glutamine. All 3 of those amino acids will bind to and activate the NMDA receptor. An antagonist to that receptor, one that we know of and we've seen in the literature quite a bit, is ketamine. And it does have some efficacy at very low dose in remitting MDD, much lower dose than what's used recreationally. GABA binds to its own class of receptors, so it's not really involved in this excitatory neurotransmission or diffusion of the excitatory neurotransmission in MDD. But it is something to keep in mind because it is a central player in MDD, but it has its own receptors. So the NMDA receptor works like this. Magnesium is normally blocking that NMDA receptor. So molecules of glutamic acid are secreted. They bind to the receptor like the NMDA or the AMPA receptor we talked about briefly a few slides ago. When that happens, this is an ionotropic effect. So AMPA receptors emit sodium ions, and that when activated results in the moderate depolarization of that membrane. Okay, that dislodges the magnesium ion. When magnesium is dislodged from the NMDA receptor, calcium flows into the neuron. Large quantities of calcium are going to enter then into that neuron, and what they're going to do is evoke a series of intracellular signaling phenomena, moving through phospholipase, phospholynyl 3 kinase activity, and a whole host of kinases and phosphatases, which can actually both alter gene expression and alter metabolism. Finally, this results in the addition of more AMPA receptors. More of them are recruited to the membrane so that you get increasing amounts of sodium, and ultimately the magnesium comes back and stops up that NMDA receptor, and the whole cycle starts over when more glutamate comes. Another artist's depiction of this, showing you some of the detail of this spanning membrane receptors. Remember, these are helical proteins that are found in the membrane of the neuron postsynaptically. So here's the AMPA receptors, here's the kainite, glutamate R5 through 7, and here are the NMDA receptors. Those are all ionotropic. They all generate a change in the electrical conductivity across that membrane. We also have a metabotropic glutamate receptor, and the metabotropic does not involve ion flux, but it does involve G protein coupled receptors. And those G protein coupled receptors have their own effector molecules intracellularly, which also can involve phospholipids, eicosanoids, as well as ceramide and sphingolipid metabolism, and in fact the cyclic AMP cascades and phosphatidylinositol cascades. Now, besides that, okay, I want you to take a step back now. All those proteins we were just looking at, for example, those proteins that are in the membrane, those receptors, those proteins, and indeed soluble enzymatic proteins, can all go through protein modification, covalent modification. Now, we all are aware of phosphorylation. That's what kinases do, protein kinases do. And phosphatases remove phosphate. But besides that, there's another major player in the requisite regulation of protein activity, changing its valence from either on or off, or kicking it to high gear, or diminishing or lowering its signal. Okay? That is acetylation of proteins, adding acetate. All right? Now, there's a protein which is an acetyltransferase, and it's got a lot of recognition in the nucleus of cells, and that's called HAT. That's histone acetyltransferase. So when you acetylate histone residues in chromatin, there's DNA bound to chromatin, and you acetylate the histones, you basically set that DNA up for a chromatin remodeling event, which will allow for more transcription. That is more global gene expression when DNA that is bound in chromatin is acetylated at certain histone lysine residues. Now, if you deacetylate that, you deacetylate those specific lysine residues on some of those histone proteins that are bound to the chromatin, what you do is you collapse or condense that chromatin. You make a structure called heterochromatin, and heterochromatin is notoriously transcriptionally deficient. So when you deacetylate chromatin at the histone level, you effectively diminish the amount of transcription of many of the transcripts that may have been activated previously. It doesn't diminish all the transcription, but it diminishes globally a lot of fresh transcription, nascent transcription, because of the heterochromatin condensation. So you understand then that these proteins called sirtuins deacetylate, so they condense chromatin, they diminish global transcription. That's the important take-home point. So Let's go back now and think about neurochemistry and MDD. Altered expression of receptor isoforms via sirtuin. So sirtuin is not simple, of course. There's at least 6 different isoforms of this protein. Sirtuin 1 inhibition. So when you inhibit Sirtuin 1, what do you get? Well, if Sirtuin 1 is a deacetylase, if you inhibit the deacetylase, that means you maintain hyperacetylation, right? So if you maintain hyperacetylation, what you get is enhanced expression of all of those glutamatergic receptors, A2, A3, and K5. You also enhance the expression, that is fresh transcription, of the metabotropic glutamate receptors. Now that was recently published in Scientific Reporter 2016, late 2016, just a few months ago. Okay, so sirtuins have a mode of impact on expression of those glutamatergic receptors. Okay, so how is that going to relate to MDD? We'll find out in a minute. Also, we get altered activities of metabolic enzymes. Some of them are metalloproteinases, actually, of metabolic enzymes via this covalent modification of acetate. So there are acetylation, deacetylation cycles. This was first described, fully described in a Cell paper back in 2006, some 10, 11 years ago. But I want you to look at that paper if you want to get a full-blown understanding of how excited people were when they found out that proteins could be acetylated and that could alter metabolism. Sirt4, that's only one of the isoforms of the acetylase, actually inhibits glutamate dehydrogenase. That is an enzyme that we recently saw. What does glutamate dehydrogenase do? Glutamate dehydrogenase converts glutamate to alpha-ketogluteric acid, right? And when you convert the alpha-ketogluteric acid, it goes on to the TCA cycle. But what else that's effectively doing is it's diminishing glutamic acid levels, okay? So Sirt4, when it deacetylates that enzyme, now you're deacetylating the enzyme itself, okay? It's going to inhibit that enzyme. Now that opposes that whole activity, the effects of calorie restriction. So people have been looking at caloric restriction for lots of things in the body, including neuropsychiatric disease. Of course, things like obesity, big and looming, type 2 diabetes, metabolic syndrome, you name it. These are all interconnected. Everything feeds back into the CNS, remember. So that it looks like Sirtuin 4, because it inhibits glutamate dehydrogenase, is going to oppose the effects of caloric restriction. Why is that? And by the way, that's in the pancreas. Now, how is that? Was that have anything to do with central nervous system? Because the pancreas secretes insulin. And insulin has a global effect on neurotransmission, both in the CNS and in the gut. And the two, of course, talk. So that's a whole other story, which I won't talk about tonight. But you can hopefully appreciate the fact that insulin, being a global hormone, will have effects like that. Now, recreational drugs, so-called recreational drugs. All drugs like this are dangerous and bad, but one of them is methamphetamine, unfortunately a very common recreational drug. What does it do? Well, it can alter the glutamatergic receptor gene expression as well. Not only do they have a direct effect on neurotransmission, okay, which we could talk about in terms of the monoamine circuit. I won't talk about tonight, but we could talk about sometime. What I want to say, it also has an epigenetic effect. What do I mean by epigenetic? Same thing I was just talking about. Acetylation, deacetylation of histones is an epigenetic phenomena. Likewise, methylation of histones and of cytosine residues and CPG islands and promoter regions of genes, specifically in genes where there are enhancer elements that are associated with fresh gene expression during chromatin remodeling. Okay? So that's an epigenetic phenomenon. Now, looks like methamphetamine has an effect on that pathway. So, for example, methamphetamines cause histone 4 hypoacetylation, and that then generates what's called meth-induced decrease in striatal glutamate receptor expression. Okay? It decreases striatal glutamate receptor expression. That's another important thing, and that was found in Biological Psychiatry, or published. Now, this does show structures, so I'm happy. All I want you to see here is that Sirtuin, this seemingly insignificant protein that was only recently discovered in the 90s, which is recent for someone like me, you can see that Sirtuin does two things. it will acetylate, it will deacetylate a protein or remove the acetate. You see there, remove the acetate on a specific lysine residue on a protein. It could be any protein, histone, for example, GDH, for example, isocitrate dehydrogenase, for example, all kinds of metabolic enzymes. But also, it generates this ADP ribose. That ADP ribose, shown here, can be used also to covalently modify proteins. So you get a process called ADP ribosylation. ADP ribosylation, therefore, is related to sirtuin activity. So sirtuin actually has two very potent biochemical effects. It deacetylates and it coordinates ADP ribosylation. Now, ADP ribosylation as a covalent modification is another entire interesting nuanced story as it relates to the regulation of metabolism, including things in the CNS. Not going to talk about that aspect. I bracketed that off. I just wanted to show you sirtuins are very potent proteins. Here's just a story for SIRT1 and how it might be associated with many diseases. SIRT1 is going to affect transcription factors. Which ones? I'll show you in a moment. That's going to cause chromatin remodeling. It's also going to be associated with DNA repair. So SIRT1 is one of these enzymes that people are looking at in aging, okay, or in neurodegeneration, such as Alzheimer's disease and Parkinson's disease and prefrontal dementia. Now, besides that, SIRT1 also has an effect on metabolism. I already mentioned neurodegenerative diseases as one of the diseases it has impact on. Cancer, because cancer, of course, is related to alterations in fuel economy in cells, therefore for cell division, and also for mutation rate because of DNA repair. DNA repair is related to sirtuin activity because DNA repair is associated with chromatin remodeling. Chromatin remodeling is associated with acetylation. Cardiovascular disease, and in fact, the entire inflammatory response. This is one of my most favored topics is inflammation and the immune system. Again, we're bracketing that off tonight. Where does caloric restriction come in at? Caloric restriction is going to impact sirtuins, AMP kinase. AMP kinase is an enzyme which specifically phosphorylates. It's actually a transcription factor by phosphorylating proteins that ultimately are associated with autophagy. Caloric restriction is also going to shut down TOR or mTOR, that's target of rapamycin, which means it's going to shut down nascent protein synthesis. And caloric restriction, of course, is going to affect insulin signaling for the obvious reasons because of the amount of glucose in circulation, right? So usually caloric restriction involves some kind of diminution of some biofuel. One of them could be sugar. Now, that all can have a negative effect that is a lowering of reproductive rate, and it can also affect resistance to infection, right? Because it's diminishing in the inflammation response. Now, those are two negatives if you want to consider down regulations. Now, it also has an effect on metabolic rate. But because it has a suppression of metabolic rate, it will not allow for cancerous cell division. And it also seems to have a mood mellowing effect in some mood disorders, including major depressive disorder and anxiety disorder. Okay. Now, a decrease, of course, in the cancer incidence is going to increase health and longevity, and an enhancement of mood and behavior by depressing negative aspects of mood and behavior can also increase the overall health and longevity of the individual. But beyond that, you also get better DNA repair, and you also get, perhaps paradoxically, an increase in the aggression anger circuitry in the brain, something we haven't talked about, but which we can certainly entertain some other time. So aggression also can favor longevity and health. Okay. Now, in the fasting liver, SIRT1 controls pathways that are responsible for upregulation of gluconeogenesis. So you're going to get more net glucose synthesis from non-carbohydrate precursors, right? This again is fasting. This is the fasting mode. However, the early stages of fasting, you get gluconeogenesis, and it's going to be turned on by two transcription factors, CREB and CRTC2. Those transcription factors are also going to enhance the transcription of SIRT1. So you can get more SIRT1. Wow, what does that look like? It looks like it's a feedback mechanism, and indeed, that's what you're going to get. So Sirtuin activation will then trigger deacetylation, because that's what that enzyme does. And you're going to get subsequent ubiquitination. And when you ubiquitinate a protein-specific lysine residues and you polyubiquitinate it, what do? Shuffle it off to the proteasome for degradation. So you degrade the transcription factors. That then lowers the expression of all those Krebs targets. And what does that do to gluconeogenesis? It tanks it, okay? Lowers it. So at the same time, Sirtuin also deacetylates but activates a peroxisomal transcription factor and FOXO transcription factors. Both of those are transcription factors which turn on genes which allow the body to do long-term fasting. And what that normally involves is lipid breakdown from the adipose sent to the liver, and then ultimately the beta oxidation pathway, which is going to allow the production of acetoacetate and beta hydroxybutyrate, which are then the ketone bodies which will keep the central nervous system going when glucose levels are suppressed. This fasting mode is considered quite a good, healthy environment for the human body, actually. And it seems to have an axial effect, a global axial effect on good mood and a good neuropsychiatric outcome in some literature that's been published, which of course needs to be vetted. So sirtuins play an important role in controlling inflammation, and that can contribute to a lot of chronic diseases, and we've already kind of talked about that. So Sirt1 and Sirt6 actually suppress the activity of a very important key activator, transcriptional activator of inflammatory response, and that's NF-kappa B. Sirt1 directly deacetylates one of the subunits of that transcription factor. It reduces the ability of the transcription factor to activate transcription of all those pro-inflammatory genes, like pro-inflammatory cytokines, okay, and enzymes like cyclooxygenase. Sirt6 also deacetylates histones at promoters for the NF-kappa B. And what happens when you deacetylate? I told you condense that chromatin. What happens when you condense the chromatin? You knock out global gene expression. Knock it out. Okay. So Sirt1 and Sirt6 may work in concert, as Sirt1 can activate Sirt6 transcription. Again, it's kind of a crosstalk between those two transcription, translational activation pathways. And overall, then, you're going to diminish inflammation. All right. Here's a paper that comes out of Physiological Review in 2012. Besides those sirtuins you find in the nucleus, they're also mitochondrial. They govern key aspects of energy metabolism, especially during prolonged fasting. So this is going to be more about a metabolic activity. Sirt3 is a major player here. Sirt3, that deacetylase, promotes the catabolism of acetate and of amino acids. So the amino acids are going to be run through the TCA cycle and ultimately can be burned in the TCA cycle for NADH production and then ATP synthesis. Okay. So this Sirt3 will deacetylate that enzyme that we've been looking at off and on throughout this talk, glutamate dehydrogenase. That enzyme converts glutamate to alpha-ketoglutarate. Okay, now we talked about Sirt4 having an effect on that enzyme. Look at Sirt3 as an opposite effect. Okay, so Sirt3, okay, deacetylates but activates glutamate dehydrogenase. Sirt4 did the opposite. So depending on the expression, which sirtuin is being expressed, which is of course tissue specific, you're going to have either an increase or a decrease in glutamic acid. So glutamic acid is there binding to the glutamatergic receptors in the CNS. This is going to have a very specific, potent effect on glutamatergic excitatory neurotransmission, where those sirtuins are expressed or induced to be expressed for something caused by something associated with caloric restriction, for example. Likewise, acetyl-CoA synthase is also deacetylated. And again, what happens is you activate that enzyme. Why is that important? Because you put acetate into play. You put acetate into play, it can go on to make ketone bodies. You can also run the TCA cycle, NADH, ATP synthesis. So what's the summary of all this? We're almost at the end. Glutamate, sirtuins on MDD. Complex interactions all over the place. On one end, something we really didn't talk about, but I'm just going to mention here, glutamic acid stimulates neuroplasticity. And so that may enhance recovery from triggers to major depressive disorder. That actually may be part of the positive healing effect, okay, increasing neuroplasticity. That's a totally different pathway for MDD. That has a lot to do with cognitive behavioral therapy and more of the psychological perspectives on how to deal with major depressive disorder. But that's something we have to keep in mind when we're thinking about the modulation of glutamic acid. We don't want to tank glutamic acid, because we do that, we may be impacting on a pathway which may be positive for MDD. So all these drugs we talk about, that's why we want to look at modulation. At the very beginning, I told you I'd talk about modulation. We don't want to talk about totally knocking something out or jacking it up. We want to modulate that. We want to modulate that specific tissue types, maybe at specific neurons. On the other hand, inhibition of those glutamatergic receptors has been shown, as I mentioned also early on, as a pharmacotherapeutic in MDD. So we need to tease out, right? We need to figure out receptor turnover, and that can have to do with that deacetylation pathway and ubiquitination and proteasomal degradation, right? Normal aging of those receptors. Also nascent expression of those receptors because of chromatin remodeling and acetylation. And also the amount of glutamate that's in the synapse. And again, that comes down into metabolism. Glutamate being metabolized, for example, by GDH. So sirtuins are on the horizon. They're on the horizon. They're right now considered druggable targets. There's 6 different forms of them. They are quite specific, as we've been seeing. I just scratched the surface there. They're also heavily researched in aging, cancer, and cardiovascular disease. So it's no wonder that they're starting to be looked at in neuropsychiatry. Okay, again, my name is Dr. Dan Guerra. I am co-founder and chief scientific officer of VerEvMed. There is our email address and there's our website. And what we do is we are scientists. And what we do as scientists, we work with you to verify published evidence in research that's been centered around biomedicine and biopharmaceutical sciences. So thank you very much for your attention. Hopefully this was a stimulating topic for you to be able to be entertained by whenever you happen to listen to it. My subsequent interactions here on YouTube will be dealing with different parameters. Some of them will not be targeted towards professional audience, but more towards the layperson client. So I'm going to move back and forth. But what we want is we want clients to come to our website and to engage with us one-on-one so that we can work with you with your specific questions. And we dig into the literature and we do for you. We communicate with you at the level that you feel comfortable with, and we help to dissect and disambiguate the literature and try to verify what's there in the evidence. So thanks a lot for your attention again, as I said, and we will see you later.